Воду не купила, думаю, холодно будет, воду эту пить.
А тут тепло, вот тут самые те комнаты, где жарко.
Да? Ну это отлично вообще, хорошо.
Все готово.
Спасибо большое. So, good afternoon, guys. Good afternoon, Sadi Parajak, Nirav Vamja. You are those who came first. Good morning or good afternoon. And I hope you have a tea or coffee in front of you. I also get some latte coffee in front of me. And or soft drink as you wish. And today we are going to listen about hepatitis viruses. So my dears, hepatitis viruses, these are very important group of viruses that gives a very large or high share of the liver pathology. This pathology is of utmost importance, and we cannot really overestimate the epidemiological importance of hepatitis virus. We have studied already some of the hepatitis viruses that were hepatitis A and hepatitis E, and these are those that can be transmitted through the gastrointestinal tract. So they are called enteric hepatitis viruses. So, and they can be enteric, or they can be passed through the food or water through GIT, because they are non-enveloped viruses and they are very resistant to the stomach acidity, to the liver enzymes, and cannot be inactivated, these intestinal enzymes, and cannot be inactivated there. So, but then today we will be discussing not all hepatitis viruses, but only those that are parenteral hepatitis viruses. But first, what are the most common features of all hepatitis viruses, if any common features can be seen? Of course, they are of very narrow tissue tropism. So they can cause infection of one single organ, that is liver. And if we have acute disease, and any of hepatitis viruses that exist may cause acute liver infection. So then we may see the very common so-called icteric symptoms, and you can see the liver damaged with the hepatitis virus. And the icteric symptoms are usually connected with the skin, mucous membranes, eyes discoloration or yellowing of them, nails, so, and also changing of some biochemical characteristics of the blood. So liver enzymes and bilirubin come out of the damaged liver cells, hepatocytes, and will change the biochemistry of the blood. So then second very important feature typical for almost all hepatitis viruses, that they are readily spread. And the problem is that the person who is infected start releasing viruses or start to be contagious before or without showing symptoms. Before, it means in the last portion of the incubation period, the individuals infected with hepatitis viruses start to be contagious. Can you imagine? They don't even know that they are infected, but they can infect the others already. So, and even if we may have a case of asymptomatic hepatitis, so still this person can be infectious or contagious without producing any symptoms. So then one more very important feature. So all hepatitis viruses are not related taxonomically. They are all distributed within the different families and within the different genera. So that's why they have different design of the nucleic acid. So different structure, different chemical compositions, and modes of replication, and they also, we may divide them according to the mode of transmission. So then, because all this is different, the specific cause, nature, and serology of the disease. What do we mean under serology? The appearance of the immunity and the types of antibodies that are produced and cell-mediated immunity, whether it is produced or not. So all these cause, nature, and serology of the diseases caused by hepatitis viruses are all differ for each virus. So now this is a table where the discovered by now hepatitis viruses are mentioned. So, and here we can see a group of RNA viruses, and there the first discovered was hepatitis A virus, discovered by Feiston 1973. And then we have hepatitis E, C, D, and G. And the only really white one DNA virus is well known, and the disease had been discovered in 1963-64 by Blumberg, and the virus had been seen by Dane. So, and then we have some variants. So we don't know whether these are the separate viruses or the variant of DNA viruses. Until now, it is not really known very well. But here we have the one of the most common hepatitis virus, which is hepatitis B virus. And it had been discovered or seen at least in 1970. So truly, this is the first discovered hepatitis virus. So, except for A and B, the rest of the hepatitis viruses, because they had been identified later on, so quite often, even in clinic, they are called non-A, non-B hepatitis viruses, to underline that they are not so common as A and B. So for the very long while, the doctors knew that there are some other viruses that cause the similar diseases, but they had not been identified for quite a long while. See, E until 1980, C until 1989. So then the G95 or so. So then they are traditionally called non-A, non-B hepatitis viruses. So regarding the hepatitis viruses, they all can be classified broadly into two different categories. I mean, those that are very well identified, not HFV and not HTTV. So the major hepatitis viruses are divided according to their way of transmission. As we said, hepatitis A and E demonstrate the fecal oral route of transmission, while the others, B, C, D, G, are called parenteral hepatitis viruses because they undergo parenteral transmission. What does it mean, parenteral transmission? This is transmission by any route, really, but not through the GIT, because they are all complex viruses within fragile envelope with glycoproteins. So that's why those fragile membrane can be very easily destroyed or inactivated. So that's why. So they will be very quickly inactivated in the GIT. So they most probably transmitted through the injuries on the skin, on mucous membranes, through sexual contact, through needle injections or whatever, but not through sexual contact, right? For some of them it's really important. So, but not through the GIT. So it means para, around, enteral, by any route other than enteral. Here you can see the distinguishing features of most common these three HBV, HCV and HDV hepatitis viruses. So the disease caused by HBV is called serum hepatitis. HCV had been discovered later on. It is called post-transfusion hepatitis. And HDV is called somewhere and quite often delta agent. It's not really virus because it is defective virus. It doesn't have a portion that is needed for infection. So that's why it is called delta agent. And among those, the only hepatitis B is double-stranded DNA, only has double-stranded DNA, genome partially double-stranded, and HCV and HDV have positive sense RNA RNA molecule as a genome. So they belong to different families. So family Hepadnavirida have just hepatitis B virus, so hepadnavirus, orthohepadna hepatitis DNA virus. So HCV, Flaviviridae family, genus Hep C virus or heparnavirus quite often. So it means hepatitis RNA virus. And the HDV is not classified, it is viroid-like particle. So they are all enveloped, but please take a look. The envelope of HBV is made up of hepatitis B surface antigen. That's how we call the surface protein of hepatitis B. Hepatitis C has two types of the surface glycoproteins, E1 and E2, envelope protein 1 and envelope protein 2. So hepatitis D virus needs to borrow the envelope protein from hepatitis B. So in fact, this is a parasite of hepatitis B virus. This is a virus that is parasite of another virus. Then regarding their size, so the HCV is of middle size, around 60 nanometers in diameter, and B and D rather small, considered to be rather small viruses. So they are all acid and ether sensitive, so they will be digested in the GIT. And the prevalence of this hepatitis B and C is very high right now, really very high. So the prevalence of the hepatitis D virus infection is lower and regional. Then a few words about their infections. We compare caused by hepatitis B, C, and D. So the routes of transmission are same. So, and please take a look. What is typical for them is the very long incubation period, up to 160, 180 days. At least a bit less for hepatitis D, but still 65 days are quite a lot. So the onset is insidious for HBV and HCV, but very acute, sharp, and severe for hepatitis D. So those two can rare cause fulminant hepatitis. So HDV may cause it with high to very high possibility. So, and then please compare the examples on what type of the infections are typical. For hepatitis B, acute disease is typical. It occurs up to 80% of people, and chronic disease in 5 to 10%. While the hepatitis C virus is well known for its ability to cause very long-term, mild, chronic diseases at the beginning, but then out of these, 20% cause cirrhosis, and 5% of those that cause cirrhosis may cause cancer. So it means it is really very mild at the beginning, but when the liver, when the inflammation persists in the liver, then it starts to cause very severe consequences. So that's also typical for hepatitis B. In 10% of chronic infection patients, the cirrhosis will be developed, and out of those, in 80% from those who develop cirrhosis, the primary hepatocellular carcinoma will be developed. So very high oncogenic potential. So hepatitis C also has an oncogenic potential, but much lower. So for hepatitis D virus, cirrhosis is typical, but then as a result, fulminant hepatitis is most typical. So, and if we take a look at I will add not only prevention but treatment, at the treatment of these diseases. So hepatitis B, very good vaccine, everyone is infected, but no good treatment. Hepatitis C virus, no vaccines available, but right now, within the last decade, the very good treatment, or the last 5 years, for example, the new targets had been found for hepatitis C replicative cycle, and very good treatment is developed. So hepatitis D virus the infection cannot be really treated very good, but vaccination against hepatitis B also protect against hepatitis D, because they have the same glycoprotein to bind with the cell surface receptor. So the blocking of this glycoprotein will cause neutralization of the virus B, both viruses B and D. So few words about the significance of that group of the viruses. So you can see here that more than 500 million people are persistently infected on Earth with hepatitis B or C and C. And if they are infected, they are at risk of having chronic hepatitis, cirrhosis, and liver cancer. So then the second very important option. These hepatitis viruses cannot be properly cultured in vitro. It means until now we don't have very good model how to grow this virus. And moreover, so we don't have good model of this virus in infected animals. So the infection of animals other than primates is really very tricky. So what is the result? So we have limits in understanding of the virus life cycle, and this is due to the lack of culture model, and we don't know very well the pathogenesis of the disease, because still we have some gaps. So why? Because the very good model is needed to study very well the way of the way of the pathogenesis. So that's why it was difficult to develop vaccines, and for hepatitis C, still we cannot do this, and it was difficult to find out the antivirals. So of course we have some progress in this. In 2014, a novel human hepatoma cell lines uh, have been established or have been designed that support the entire life cycle of both hepatitis B and C viruses. And this cell line provides a powerful tool for addressing the virus life cycle and the development of the antivirals and vaccines. But by the way, even in this very good cell line, not always the infectious virions are produced in the cells. That is the problem. So virus replicates, but it fails to produce completely done virions that can infect other cells. So that is the problem with this. So that's why until now we are not using culture of these viruses in the clinical routine diagnosis, and maybe we will never use it because it is very difficult, extremely uh, costly, and takes time. So we use different ways for diagnosis, and we'll discuss them. So we start with hepatitis B virus, our description, because of course still it is most common and incidence of the disease is highest. So it usually infects the liver, but also may multiply in kidney and pancreas of humans and Japanese. So it is small DNA virus, and it has an envelope made up of lipid bilayer, right, and the hepatitis B surface antigen. It has a unique genome. It is very small, 3.2 kilobases base pairs, right, circular, partly double-stranded. What does it mean, partly double-stranded? It means the negative sense DNA in double-stranded DNA is complete and positive sense DNA is not complete. And also they have unique enzyme inside. It is polymerase with reverse transcriptase activity. So it means it can form the DNA based in RNA as a template. You know that just only one type of the viruses, except for hepatitis B, have this ability. These are the HIV viruses, and altogether all viruses from retroviridae family, because except for HIV we have some other viruses there. So based on sequence comparing, hepatitis B virus can be classified into, I forget. So this is the yellow one. This is major core antigen, that is called hepatitis B core antigen, HBC antigen. From the very beginning you should ask me why you call these proteins refer to the viral proteins as antigens. And the answer is because they are all proteins and they are all immunogenic, and combinations of these proteins that are found in the human blood and antibodies against these proteins are used as so-called serological markers for differential diagnosis of this disease and for the identification of the stage of the diseases. So balance between these proteins and the antibodies against these proteins may be used also as a prognostic sign to understand what is the most probable outcome of the case that we have. So that's why regarding their immunogenicity and importance in the immunity development and diagnosis, we call them antigens. So you please do it in the same way. So, and based on the base pair sequences, the hepatitis B viruses can be classified into 8 genotypes, and each genotype has a distinct geographic distribution. So now let's take a closer look at the structure of what we see of the virus and some virion particle that we see in the blood. So if we take the patient's blood serum and take a look under the electron microscope. So then we will see 3 types of particles. You can see these 1, 2, and 3. 2 and 3 are, and they are called, seen here, 2, and this is a 3. These are subvirional particles. What does it mean? It means these are not infectious virions. These are particles that are made up of HBs surface protein or antigen of hepatitis B. So they are empty, they don't have the core and don't have the nucleic acid inside. So they are not infectious. They're just particles that are inserted or made up of glycoprotein that are inserted into host-derived lipids without any genome. So they are not infectious. And that's, you see here them very well, right? They are made up. So usually they have 22 millimeters in diameter, and one is spherical and the other one is quite long. It can be up to 17 nanometers in length. So come back. But the most important particle is so-called Dane particle. This is infectious hepatitis B virion. The virion is of 42 nanometers in diameter, and you see the lipid bilayer, HBs antigen, HBc antigen, polymerase covalently bound or fixed with the nucleic acid, with the genome. So, and if you take a look at the cross section, we can see also here the hepatitis B, C or core antigen there. So there we also have the very small minute amount of HB, but then we have the HB is also core antigen. But we have really totally different processing of the HBc antigen and HBe antigen in the infected cell. The thing is that hepatitis B core antigen will be involved in the virion development, right? In virion so-called morphogenesis. So the HBc antigen is not really released into the bloodstream. So we should not use it itself, this protein, as a serological marker. While the HBe antigen will be released into the serum, and that is the unique way of its processing. So it can be found there And HB antigen is a very important serological marker that will tell about the high high level of virus replication in the body and high infectivity of or contagiousness of the patient. So the structure of the surface glycoprotein is quite sophisticated. So it is made up of small glycoprotein, medium glycoprotein, and like you measure the size of the tissues, right? S, M, and L glycoproteins. So, and they are essential for the virion assembly. And the combination of these antigens that are responsible for surface glycoproteins result in the subtypes development of hepatitis B virus. For example, we have these glycoproteins. They may contain group-specific antigen, typical for all of them, but also type-specific antigens. So regarding those antigens, we may variations of it. So the structure is unique, so do the replicative cycle. So now we discuss the replicative cycle because that is the example of one of the most sophisticated way of virus replication. So first we have some unique features of the replication cycle. So first, hepatitis B virus has very strong tropism for the liver. And it uses not so far ago, the specific receptor is found to which this virus binds. And it is called sodium taurocholate co-transporting polypeptide, NTCP. And there's a major cellular receptor, and because the cellular receptor is unique for hepatocytes, so the virus multiply in hepatocytes only. Then what is really unusual? The hepatitis B virus is DNA virus, but it replicates through the RNA intermediate. So this is like mirror reflection of the HIV. HIV virus is RNA virus that replicate through DNA intermediate, right? They both unique. So then hepatitis B virus produce because it causes overproduction of hepatitis B surface antigens. So then they will be secreted and to form the HBs containing subvirional particles, and those particles may act as antigenic decoy for antibodies produced in a body. And by the way, so those particles together with antibodies produced against HBs antigen may form very large so-called immune complexes. So we may have complications of hepatitis B infection associated with this type 3 immunopathological reaction, immune complexes hypersensitivity. I think you are not studying immunology, but you studied pathophysiology, and I think you have passed this already in the pathophysiology. So this is a typical type 3 hypersensitivity reaction. So apart from those, we also may have minor hepatitis B virus, so-called X protein. And it is essential for virus infection and has been implicated in the development of the liver cancer because this X protein is a transactivator of transcription. So you can see here the specific NTCP receptor. So, and it acts as a cellular receptor for hepatitis B virus and for its satellite hepatitis D virus covered with the HBs antigen. This is a major determinant for liver tropism and entry. So then, due to the entry, you can see here, the virus fuses its envelope with the surface of the infected cell. And the icosahedral core will enter the cell, as you see here. Then the virus is decapsidated, and the genomic DNA should be transferred into the nucleus. But then, okay, it doesn't work. Why? My dears. It's stuck, sorry. Okay. So, and then the first thing that happens, that you can see here, that the first thing that happens as soon as the virus enters, it should be complete synthesis over the positive sense RNA. That is shorter than the negative one. So first the repairing of the DNA molecule, and then with the repaired DNA molecule, the capsid will be deassembled in the nucleic membrane, and the nucleic acid will enter the nucleus. So then in the nucleus, repaired DNA start to be supercoiled, and these supercoiled viral DNA molecules start to form so-called, can you see here, CCC DNA molecules, circular, covalently closed, double-stranded DNA molecules. And by the way, the abnormal amount of these CCC molecules will be formed in the cell. So those 3C DNA molecules have a very long half-life. And interestingly, they are usually not integrated in the viral genome. And while they occur in non-integrated state, it is believed that 3C DNA molecules can be transported to the neighboring cells for infection. So they can be infectious if they are transmitted directly to the neighboring cells. Then the next step in replication is when these DNA molecules are transcribed into mRNAs. So see, they undergo transcription. And then mRNAs will be transported from the nucleus to the cytoplasm to perform translation. So the core protein will be translated and the polymerase will be translated. So, and these two types of proteins may start forming the new cores and assemble with the transcribed with the RNA. My dears, that is interestingly, that pre-genomic RNA will be encapsulated. See, this is DNA virus, but RNA molecule will be enclosed into the capsid. So then, as soon as this core with the nucleic acid is formed, then this RNA will be immediately transcribed with the help of RT, reverse transcriptase, that is the polymerase of hepatitis B virus, into DNA. So, and because it is positive sense pregenomic RNA, the minus sense DNA will be completely built in the capsid. Then the polymerase also has the rhRNase activity. It means that the pregenomic RNA will be immediately destroyed, and then using negative sense DNA as a template, the polymerase will start producing a positive sense copy of the DNA. So positive sense strand DNA. So while it is ongoing, so then the virus start budding from the Golgi and endoplasmic reticulum, where the surface glycoproteins are already prepared, hepatitis C, hepatitis B surface antigen. And then those particles that obtain the membrane from the Golgi, they will be transported and released through the exocytosis. So these infectious virus particles will be produced. But then usually it will be not enough time for polymerase to build up a complete copy of positive sense DNA. So it will be released partially double-stranded. So, but then a parallel process that occurs is the overproduction of hepatitis B surface antigen, and from the membranes and the glycoproteins, the subvirional particles or subviral particles will be produced and released by exocytosis. But also we have the process of exacerbation of the infection. When the cores with partially double-stranded DNA molecule is produced, then those cores also again may be decapsidated on this nuclear membranes, and the nucleic acid genome may enter the nucleus of the cell. So again, CCC DNA copies will be produced. I hope you understand the whole thing. If you would like to see to take a picture of the slides, so then they are here. So, this is the whole replicative cycle. And we're supposed to take a look at the targets in this replicative cycle that may help in treatment of hepatitis B infection. Because this is DNA virus, it have lower sensitivity to interferon. Then to prevent the virus, the so-called anti-CP blockers can be used, or anti-CP inhibitors. So these are mircludex, cyclosporin, and the inhibitors of the anti-CP can be used on the beginning stage of the infection. Then the also good stage for the inhibition of the virus replication is inhibition of the reverse transcriptase. But this is really very tricky in hepatitis B virus. This strategy works very well in HIV virus polymerase reverse transcriptase inhibition, but it works not so good in hepatitis B virus polymerase inhibition. So nucleoside analogs can be used, and they are used in treatment of hepatitis B infection. But by the way, they are not very effective. So by now we have just two targets, the entry and the reverse transcriptase activity. So then, one more thing about the replicative cycle. The integration of the hepatitis B genome may happen into the host cell DNA. So if we come back to here, see, so sometimes not just 3C DNA are produced, but then the DNA may enter the nucleus of the cell. And when it happens, the long-term infection occurs with hepatitis B, and as a result, the development of hepatocellular carcinoma. So the transformation of the cells occurs mainly with the help of X protein that acts, as I said, as transactivator of transcription of not only viral genes but also cellular genes. So it turns on the transcription, and as a result the overproduction of cellular proteins, and this all stimulates the cell growth. That's how it may lead to the neoplasia. So now pathogenesis of hepatitis B infection. So you can see here that the all body fluids are infectious. And this is vaginal secretion, blood, semen, saliva, mother's milk. So with this, the virus can be transmitted horizontally, but also through the bloodstream. The virus can be transmitted vertically from mother to child. So neonates can be infected through the, even fetus can be infected, but also neonates can be infected with the hepatitis B. And as soon as the virus enters the body, it enters the bloodstream through the injuries on mucous membranes and many ways. Then when it circulates in the bloodstream, usually it circulates until it reaches the liver. So, and if antibodies are present in the blood, they can prevent spreading of the virus and binding of the virus to the liver cells, right? Receptors. So then the presence of antibodies can block the infection. And also when the liver is infected and the antigen is released, so the antibodies may form the immune complexes with the antigen and antibody. So that will result in immune complexes diseases. That's a typical type 3. So glomerulonephritis and arthritis. Then, if cell-mediated immunity is very strong, it can prevent the symptoms development and they may lead to the resolution of the infection. If not, then the viruses will be released by liver. We have viremia. As a result of viremia, on all fluids of the body will be infected. So that's again the virus can be shed from the body. So if we take a look at the disease, we take a look here. Mainly, the character of disease depends very much on the person's immune response to infection. So that really determine whether the disease will be acute or chronic, symptomatic or asymptomatic. Let's take a look at this. So if we have high dose of infection, a lot of cells are infected, a lot of antigens are produced, and very high immune response is developed, including effective cell-mediated immune response. It will lead to liver damage, right? Because the cell-mediated immune response is involved in killing of the virus-infected cells. But by killing virus-infected cells, they cause the liver damage, but liver may regenerate within half a year at least, right? So, but then together with this, it will lead to virus elimination from the liver. So what kind of disease we have? Acute disease with quite severe symptoms, with jaundice, with the release of liver enzymes into the blood, but then the outcome of this disease will be most probably resolution of the disease. That's one type. The second type, where not so many antigens or the lower load of the antigen enters. So So very few cells are infected. Not so much antigens are produced. So the virus established step by step the persistent infection. So because the load of antigen is not so high, the limited cell-mediated immunity is produced. So some cells may be affected, but mainly the liver will keep virus-infected cells as a persistency. That will be the persistent infection. What will be the result? Very mild symptoms, no liver damage. So, no very or a bit elevated liver enzymes in the body, no icteric phase, usually no jaundice. So, the very mild symptoms, but then this is the chronic infection in liver that causes cirrhosis, or in some cases it may result in primary hepatocellular carcinoma. That when we have chronic disease. So then we mentioned that delta agent or hepatitis D virus cannot cause disease alone. It usually causes disease along with the hepatitis B virus. So it may cause co-infection when these two viruses travel together, or super-infection when hepatitis D come over the patient with already established hepatitis B infection. So most probable result is fulminant hepatitis or with liver necrosis and very severe symptoms, and most probably the disease is lethal. Or it still may progress to primary hepatocellular carcinoma or cirrhosis. That is the result of pathogenesis. We have discussed it. So, what exactly features of the virus may lead to chronic infections? We blame the 3cc molecules because they may occur in the nucleus of the hepatocytes, and also local tolerance to HBV infection also may participate in the development of the chronic disease. If we take a look at the global prevalence, see, you and us, we live here in the world where we have between 2 to 5% of hepatitis B infected individuals. That's, by the way, is very high. So, but then we may have it even extremely high when more than 5% of population is infected. Who are at risk? My dears, if you have a medical student, because it may have contact with patient's blood, so that is drug user, that has multiple sex with multiple partners, or this is the person of triple risk, okay? So, and if you like tattoo and piercing, that will be tetra risk. So that's why, my dears, so you should prevent this high risk of the infection. But I think mostly the medical students are already fully vaccinated against this disease. Maybe you are not at risk at all. So, clinical findings. So, usually the incubation period is very long, right? Then the prodromal period happens, where we may have fever, malaise, anorexia, nausea, vomiting, abdominal discomfort, and chills. Then the icteric symptoms occur in the acute disease with jaundice, dark urine, and pale stool. So, and then usually we have recovery period, the decline in the fever and renewed hepatitis, but it may take a very long while. So, fulminant hepatitis is more severe symptom, but it occurs very rare with hepatitis B. So then, as a result of immune complexes formation, hypersensitivity reactions may cause fever, necrotizing vasculitis, polyarthritis, rash, and glomerulonephritis. These are all the symptoms that we mentioned for type 3 hypersensitivity reaction. Chronic infection usually is very mild or inapparent or asymptomatic. So, but then if we check the biochemistry of the blood, we may see the elevated liver enzymes level, but then within time, the cirrhosis will be developed in the patients and liver failure. So these people are those people that are chronically infected with hepatitis B are at risk of having fulminant disease when become co-infected with hepatitis D virus. So, and also primary hepatocellular carcinoma may be developed, but that's not a very speed or rapid process. It takes usually basically from 10 years to several decades to develop primary hepatocellular carcinoma. How do we develop diagnosis? Now we come up again to what I said at the very beginning. We have to find serological markers of the disease. Those are used for differential diagnosis. My dears, clinical symptoms and liver enzymes, the presence of liver enzymes in the blood, just may tell us that the liver is damaged, but not really used in differential diagnosis. So, usually out of the viral proteins, we may use as a serological markers, we may use HBs antigen because it is the first one secreted into the blood, HBe antigen because it is also secreted into the blood, and tell us HBe antigen will tell us mainly that we have the presence of infectious virus in the body. So, but then antibodies against hepatitis B surface antigen are also used. But then, do you remember that those two may form a complex, HBs antigen and its antibodies? So, this complex will not be seen by regular ELISA tests. For them, we need to perform a special ELISA test. So, and then hepatitis B core antigen is not released into the bloodstream, but because it is a major protein, high level of antibodies will be produced. Usually IgM antibodies against hepatitis B core antigen are very important in the differentiation of the acute hepatitis B infection. So, and here you can see the time course of the disease and the appearance of the serological markers within the acute disease and within the chronic infection, right? So, within the acute disease, we can see a lot of hepatitis B DNA by polymerase chain reaction. Even within the incubation period, sometimes before. See, jaundice is here and the hepatitis B DNA is there already. So then, hepatitis B antigen will appear in the blood before and within the icteric stage when we have jaundice. And then we rather see anti-HB antibodies than HBe antigen itself. So it will disappear from the blood soon. Then hepatitis B surface antigen appears and disappears. This is the window period. Why? Because the detecting of the hepatitis B antigen and antibody is difficult. Why? Because the immune complexes are formed. So within the chronic infection, it is characterized by continuing HBe antigen. See, there is no anti-HB antibodies formed very well, right? So then, before, within, and after the jaundice. Then we have the HBs antigen present in the blood usually. And also we may have the very high total anti-HBc antibodies, but that is the same with the acute infection. But then the anti-IgM against HBc will disappear quite quickly from both this and that. And here we also have lack of anti-HBs antibody because they form immune complexes. So, one more question is whether we always need to treat hepatitis viruses in patients. No. If we have acute HBV infection, so the liver is damaged already, we don't need, and the immunity is working hard to get rid of the virus, to eliminate the virus. So we won't help with antivirals. The only treatment is discussed when we have chronic hepatitis B virus. And there are several possibilities. So, and the only treatment supposed to be performed when the number of the when the amount of the viral DNA is very high and ILT are normal and the liver biopsy considered, then we have moderate or severe histological damage, then we perform treatment. So, but if no mild histological disease is seen, so no treatment is performed. Then one more way, if we have very high level of the enzyme, that tells us about the uh, about the process over the um, liver damage, then we perform treatment. If the liver enzymes are very low, so we won't perform treatment. But this is rather clinical, so we can go for the treatment. What kind of treatment? So, interferon treatment, these approved drugs, so the conventional pegylated and paginated. So they are used in combined antiviral therapy and immunomodulating effects. And also nucleoside analogs that affect the reverse transcriptase. These are reverse transcriptase inhibitors. So that has direct antiviral effect, but still the treatment is not very effective. It cannot help to eliminate the persistency of the virus in liver. It may just help to inhibit the replication of the virus. So, my dears, hepatitis B can be prevented by injected hepatitis B immune globulin. They may be administered as a post-exposure prophylaxis or post-exposure treatment to the newborn babies or those who were exposed. So then, to reduce the risk of transmitting through the blood, we may usually the screening of the donated blood is performed. So then, so I would like to draw your attention to the vaccination. I hope now all of you are vaccinated. Otherwise, you have to perform this vaccination as soon as possible. Usually, this is a subunit vaccine that is made. It is called recombinant because it is made by genetic engineering when a plasmid containing surface gene for hepatitis B surface antigen is cloned into the plasmid, and plasmid is cloned into the yeast Saccharomyces. When we culture them in large volume over the liquid medium. So the HBs antigen will be expressed and produced and released into the medium. Then we take medium, purified, concentrate the HBs antigen from there. So we use it as the antigen in subunit vaccine. That's it, and I would draw some attention to the hepatitis C virus because it's also very important. Okay, it belongs to Flaviviridae family, and the genus is called Hepativirus or somewhere I've read Heparnavirus, hepatitis RNA virus. So then you can see the structure. It is covered with the envelope with one type of spike made up of two dimers, E1 and E2. The virion is spherical, there's a middle size 60 nanometers, and inside it has a single-stranded positive sense RNA genome, and according to this structure, we may distinguish 6 genotypes. Here we have icosahedral capsid made up of capsid protein. So this blue one is lipid bilayer covered with the E1 and 2 glycoproteins. You see, it's very simple. So, but then around 200 million individuals are infected worldwide with the hepatitis C virus. And this represents 3% of the world population. And out of them, 3 to 5 millions in the United States. According to the WHO, the regional office in the Middle East, Egypt has the highest prevalence in the world, with around 7% over the prevalence in adults. And also this picture demonstrates the distribution of the serotypes. And I think you can see here that hepatitis C genotype 1 virus is the most common almost everywhere. Can you see? Everywhere, maybe except for Africa only. So what we see in Africa, we have genotypes 4, the very important, and genotype 5 is endemic in south part of the Africa. So then, apart from these, in Europe, we may see genotype 3. You can see it in Russia, here and there, in America, in both Americas, and also here very much, right? And also the rest is not so common. So, structure of the virion and the viral nucleic acid. So, the nucleic acid, genomic RNA, is positive sense RNA with a pin on one side that acts as the 5' cap. So, and here we have just very few structural proteins. This is a core protein and 2 envelope glycoproteins. And that's it, my dears. So the rest are non-structural proteins. So with the example of P7, this is the ion channel protein, also happen in the virion. So then the transcription and translation occurs of the whole genomic RNA, right? So, and then the cleavage of the polyprotein first will be done by cellular peptidases or proteases, and then it is performed by viral proteases. And this is a sort of autocleavage. because viral proteases NS2 and 3 work as proteases that break the polyprotein into 10 functional subunits. And also the very important non-structural proteins, for example, NSP A and B, mainly B subunit, may make the RNA-dependent RNA polymerase that is necessary to combine with NSPA protein, and they form the transcriptional complex. So, replicative cycle. What kind of problem do we have with hepatitis C virus? You should ask me. Why it is covered by some bubble? I don't know. Right? So see, this is a virus and usually it is covered by low density lipids of the cell, cellular lipids. So that's why maybe one of the explanations why this virus is low immunogenic, because it is not really very well recognized because it is covered of lipoproteins, human low density proteins, lipoproteins. So, and then they form so-called the virion on lipoproteins. So they call so-called hybrid lipoviral particles. This is lipoviral particles. So these are viral particles covered with lipids. And you can see here that the scavenger receptor class 1 type, this is scavenger receptor B1, is usually important in the lipid trafficking, and the second one is CD81. So those receptors are used as the critical receptor for virus entry, and it's understandable because these are for lipid traffic. So the lipoviral particle will bind with this receptor. So it enters with the help of these receptors by receptor-mediated endocytosis. And then here we have the endocytosis into the clathrin-mediated, clathrin-covered particles. And then we have the uncoating and the nucleocapsid will be released into the cytosol. And the viral genomic RNA is infectious by itself, so it will be sent to the ribosomes for translation. And as a result of translation, the long polyprotein will be produced, and the polyprotein is supposed to be proteolytically cleaved, and here you can see a sort of cleavage and then replication of the viral genome occurs through the minor sense intermediate. So then it is important to understand that within the replication, it happens in the so-called membranous web. So the replication complex will be formed there in the part of the cytoplasm. Then the viral assembly occurs and the release by exocytosis. This virus is not cytolytic. By the way, so do hepatitis B virus. They are both non-cytolytic. So the infectious virus particles will be released and then later on covered with the lipoproteins of the human origin. So the hepatitis C spread by the same routes as hepatitis B. So, but the transfer through the sexual contact is really extremely rare. So this 3 to 5% can be transmitted sexually, mainly by the blood, needles, and perinatally also common. So viremia can be detected within 3 weeks of the transmission. And the viremia may last for a very long while in people with acute infection, or even longer with the people with a persistent infection. So the acute hepatitis C virus is similar to that of any hepatitis, but it is extremely rare with hepatitis C. So fulminant hepatitis is also very rare, and the virus can cause fatal disease, but very rare. So most probably it causes chronic hepatitis. So why we have this? So first, because it is weakly immunogenic, then it replicates very slow, so the antigen load is not enough to evoke very strong immune response. So that's why chronic infection occurs in 80% of individuals. And hepatitis C develops persistent hepatitis that may lead to cirrhosis after 9 to 30 years. So, and cirrhosis happens in 20% of chronic illnesses. And then we have liver failure in 20 of those who have cirrhosis. And then hepatitis C is considered to be oncogenic. But then what is the way of oncogenesis? This is RNA virus that it cannot enter the genome, because RNA never enters DNA, never can combine with DNA like in so that is the problem. So it is believed that the chronic inflammatory response that is established in the liver cells is the positive, is the possible way for oncogenic transformation of the cells. So how do we perform lab diagnosis of hepatitis C infection? So the serological assays can be useful, and usually in chronic but not acute disease. But really, serological assay is not very sensitive, because first, seroconversion may occur very late, within 30 weeks of infection, and the antibodies level may be very low. So usually we use NS4 protein as the one antibody against which will be used in ELISA test. But without any doubts, the best way to diagnose hepatitis C infection is polymerase chain reaction. This is the best indicator of the disease, and also it allows us to detect the presence of the infection and to differentiate, to perform the differential diagnosis, even with people without antibodies. And you can see that a decade before, the only alpha interferon had been considered as a good treatment to prevent the cirrhosis and to prevent the hepatocellular carcinoma. But now the combination therapy of not only alpha interferon and ribavirin is used. So we have several development of the several generations over the antiviral drugs, and the first generation were boceprevir and telaprevir. These both are potent protease inhibitors, viral protease inhibitors. So, and they had been licensed in 2011. And the second generation is sofosbuvir, and this is a nucleotide analog of the cellular nucleotide that inhibits the hepatitis C polymerase. So, and after ingestion, it rapidly will be activated by cellular protease and taken up by hepatocytes and incorporated into the RNA during the activity of RNA polymerase, and the elongated RNA chain elongation of the RNA molecule will be terminated. And in 2013, it was approved for treatment in combination with the pegylated interferon and ribavirin. So, and one more drug that affect another protease, C-meprevir, is used. So we usually target the protease inhibitors, right? Oh, sorry guys, these are polymerase inhibitors. Two targets are used, right? C. Polymerase inhibitors and polymerase inhibitors and protease inhibitors. That gives us, ability to treat the hepatitis C infection. And by the way, so it was it was claimed that HCV cure is developed and can be promised. They promise that it will cure 95 to 100% of patients, and that is very promising treatment. But the vaccines are not available for hepatitis C. So good treatment, but no vaccination. Few words about hepatitis D virus. So this is small RNA delta agent. So it is unclassified because this is defective virus. So the virion is spherical, and it has an envelope made up of hepatitis B surface antigen. The virus is acid sensitive because it is enveloped, and the prevalence is low, or in some regions it can be high, but endemically. So hepatitis D has an envelope made up of HBs antigen. You can see it here, right? So on the cross section, and the only delta antigen is found here. So please don't confuse with Dane particle. This is D antigen, by which we can recognize it is a unique for hepatitis D virus. And here you can see the positive sense RNA. So it spreads by the same routes as hepatitis B virus. And damage to the liver is a result of the direct cytotoxicity, my dears. When we compare hepatitis B, C, and D viruses, B and C are not cytolytic, and D is cytolytic. So, and because it has direct cytotoxicity, when we have a combination of hepatitis D and hepatitis B, so then it is very serious combination of the viruses. It increases the severity of the disease. So, and the fulminant hepatitis is a very common result of this doubled infection. So the fulminant hepatitis with the hepatitis D lead to hepatic encephalopathy with the altered mental capacity and massive hepatic necrosis. And combination of these viruses give 80% fatal infection. So how do we perform lab diagnosis of this disease? So the delta antigen can be found by ELISA, or antibody against delta antigen can be found by can be performed by ELISA. So radioimmunoassay procedures are not so common right now because it's very difficult to utilize the waste containing isotopes. So that's why they are mainly replaced by ELISA tests. We don't have specific treatment of hepatitis D virus. And the prevention is done using vaccination with hepatitis D. What we by now know about hepatitis G virus. It is RNA virus and it also belongs to the Flaviviridae family, but the genus is unnamed yet. It is spherical and very similar with that of hepatitis C virus, 60 nanometers in diameter, envelope, single-stranded RNA, ether-sensitive, and prevalence is really moderate. So we believe that hepatitis G virus causes hepatitis G. It is transmitted parenterally. It can be identified only by polymerase chain reaction. By now, other tests developed ELISA, but they are not effective and they are not sensitive enough. So the capability of hepatitis G virus to cause fulminant or fatal disease is not known yet. So we don't have enough cases to be very certain about these modes of the diseases. So we don't know really how about the chronic diseases. We don't know about oncogenicity and vaccines are not available. So not so much about hepatitis G we know by now, but with this we may finish our today's lecture. We worked hard without any break. And I would like to announce that next week is a normal classes, but the week after the next week we start with the checkpoint 1 for those who stay in the Simferopol and started the year with the offline study. Please, the week after the next week is a control class in bacteriology, the first checkpoint. I hope everyone will hear my announcement, okay? So guys, have a nice weekend. Start studying bacteriology. See you in classes or week after the next week when we have HIV virus. So the next lecture is about HIV viruses. By the way, there are only two left, HIV viruses and the last one is coronaviruses. You are invited. Goodbye.